So in today's class, I will try to talk about, in today's class I will be talking about uh, structure of DNA, RNA, and so why DNA is what is the structure of DNA, RNA, and Watson and Crick's structure of DNA and RNA, and all these things. Uh, I'll present my screen now. Acha. First, let me go into details on what your molecular biology syllabus is. So, in the molecular biology syllabus, you have on one side the first unit is nucleic acid. In this nucleic acid, you study about the salient features of DNA and RNA. That is, you have to study about the structure of DNA, RNA, how a DNA, RNA molecule is, and you have to study about Watson and Crick model of DNA. You have to study this. In the next unit, you study about DNA replication. There DNA replication in prokaryotes and eukaryotes, DNA polymerase, chromosomes, DNA priming, replication of circular and linear double-stranded DNA, replication of telomeres. Then you study about transcription, RNA polymerase and the transcription unit, mechanism of transcription in prokaryotes and eukaryotes, transcription factors. Then you have studied translation, where you study the genetic code and degeneracy of the genetic code, wobble hypothesis, mechanism of protein synthesis in prokaryotes. Fortunately, you don't have to study this in eukaryotes. Then you have post-transcriptional modification and processing of eukaryotic RNA, concept of split genes, concept of introns and exons, splicing mechanism, alternative splicing, exon shuffling, RNA editing, and all these things. Then gene regulation in eukaryotes and prokaryotes, and you have DNA repair mechanisms and cancers and regulatory RNA. So this is it looks very heavy, but it is not a very heavy syllabus. So today I will essentially revise this unit in today's class. I will try to revise this unit. Before I go to this, you must be clear about this. So you all have a basic idea about the structure of DNA and RNA, but today I will just explain from this is iGenetics by Russell. This book is the iGenetics by Russell's iGenetic is one of my favorite books, and it's although it's iGenetics, but this is the best book of molecular biology. I have got the PDF. I will send it to you. So I don't think you need right now, since you are not getting that book in the market, you don't need to spend money to buy these books. Have a good PDF. You can print out the chapters you need if you require. But I would say that this book is just the book, only book you can read. It is good for your competitive exams as well as for other exams also. Now, let us study about the composition and structure of DNA and RNA. As we know that DNA and RNA essentially they exist as monomers, which are joined, one monomer joined together to another unit. These are monomers. Each individual circle is a monomer. It is joined individually and they form a polymer. In the case when you talk about nucleotides, it is polynucleotides. And when you talk about proteins, then you call it polypeptides. So each unit is in protein is an amino acid, and in DNA or RNA it is a nucleotide. So the monomers which make up DNA, RNA are known as nucleotides. So what does a nucleotide consist of? It consists of a pentose 5 carbon sugar, a nitrogenous base, it is also called a base, and a phosphate group. Okay. Uh, in DNA, the pentose sugar is deoxyribose, and in RNA, it is ribose. So let us look at the structure in more details. Just one thing I would like to tell you people is that whenever you are watching the on a mobile, always keep it in a side horizontal pose, not a vertical pose. Whenever you are watching, don't see your classrooms like this in a horizontal pose. Always keep it in this pose. Okay, so this is the structure of deoxyribose sugar, the sugar in DNA, and this is the structure of ribose in RNA. So as you know, this is the first carbon, this is the first carbon over here, 1 prime, this is the 2 prime, this is the 3 prime, this is the 4 prime, and this is the 5 prime. So you have 1, you have 2, you have 3, you have 4, and you have 5. So this is a deoxyribose sugar. And in this deoxyribose sugar, in the 2 prime carbon, this is the 2 prime carbon, you find only a H atom is attached, and hydrogen is attached over here. And this is the ribose sugar. The ribose sugar looks the same. You have the 1 prime carbon, the 2 prime carbon, the 3 prime carbon, the 4 prime carbon, and the 5 prime carbon. And in all of them, in the 2 prime carbon, instead of H, you have an OH group attached. So this is essentially how the sugar is there. So as I mentioned before, what you have is a sugar, a pentose sugar. These are the pentose sugar, the deoxyribose and ribose. You have the nitrogenous base. Now I will come to the nitrogenous base, and you have the phosphate group. So let us go to the nitrogenous base now. In a nucleotide, the term is, we are talking about nucleotide. NUC, nucleotide. We are talking about a nucleotide. Nucleotide, as I mentioned again, it contains of a pentose or a 5 carbon sugar, a nitrogenous base, and a phosphate group. In DNA, the sugar is deoxyribose. In RNA, it is ribose. Now let us go to the nitrogenous base. Nitrogenous base are compounds. They can be purines, they are known as purines, or they can be pyrimidines. They can be purines or they can be pyrimidines. Purine has got a double ring structure. As you can see, this is one ring and this is the other ring. and pyrimidines have got a single ring structure. This is the parent compound. So what are the parent compounds? Like all modifications of a purine gives the parent compound. So the parent compounds are, for in the case of purines, first is adenine. In this structure you have a NH2 group attached to the 6th carbon, and that is adenine. In guanine, instead of this NH2 you have a double bond oxygen over here, and the NH2 group is shifted to the 2' carbon. This is the structure of guanine. Essentially the other parts, they look the same. This is the cytosine molecule. You have in the cytosine molecule, you have a NH2 group at the 4' carbon. You have in uracil molecule, you have, it is only found in RNA. It is not found in DNA. And similarly thymine, it is only found in DNA. They are the same. Thymine and uracil are more or less the same, except only one exception, that is it has got a 5' methyl group. So on uracil, if you have a 5' methyl group over here, it becomes a thymine molecule. It is found in DNA. So this is how the structure of DNA looks like. DNA and RNA, the bases look like. And now let us go to the structure of a DNA and RNA as a nucleotide as a whole. Let us look at the structure of the nucleotide as a whole. So you can find that you have the base. In this example, the case an adenine base is attached from the smaller ring. This is how the base is attached to the sugar, and then you have the sugar. This is a DNA deoxyribose, you can see from here, and from the 5' end, from this is the 5' carbon. This, as I mentioned, in the 5' carbon you have a phosphate group, that is OPO4, OPO3, OPO3 2-. Always you see that it's written as OPO3 2-. So this is the this is the first minus, so minus the first minus, and this is the 2-, the second minus, and this is the O, and this is PO3. So this is the essentially the structure of a nucleotide.
एक बार बोल बन जाएगा था.
अच्छा. So for that, let me wipe it once more. So in a nucleotide, what do you find in a nucleotide? You find a base. In this case, you can find the base to be adenine. Find the base to be adenine. Just hold on, I am erasing this part. Okay. So you have the base adenine. It is attached to the sugar. This is a deoxy. This is the since it does not have a O group over here. It's only H. So this is a deoxyribose sugar, and since it is known as a nucleoside, base plus sugar is known as a nucleoside. So the nucleoside here is deoxyadenosine, since the base is adenine and this the sugar is deoxyribose, and so it is deoxyadenosine, and to that is attached a phosphate group. This is the phosphate group which is attached. Phosphate group, normally you will see that it is written as O PO3 2 minus. So this is the first minus, this is the second minus, therefore it is 2 minus, and this is O and this is the PO3. ঠিক আছে?
হ্যাঁ হ্যাঁ স্যার, আমি শুনতে পাইনি তখন।
আচ্ছা আচ্ছা, ওকে ওকে। So this is the nucleotide. When you add the phosphate group, the nucleoside becomes a nucleotide. Now you call this as a deoxyadenosine 5' monophosphate. Since this is the 5' sugar, 5' carbon. This is the 5' carbon. This is the 5' carbon. Therefore, this end is the 5' end, and this is here. This is the 3' carbon, and so this end is known as the 3' end. I would advise you people to read from this PDF which I will be sending from this only, because this I am explaining this concept from this book. You read it from this book and your concept will be clear. This is the best book I could find in molecular biology to understand the concepts of molecular biology. Very simple language, very beautiful figures, and concise and very conceptual. So this is how it looks. And now let us look at how a polypeptide DNA polypeptide chain looks like. This whole adenine is not drawn. In this place an A is attached, okay, and this is the deoxyribose sugar, and on the 5' end, this is the 5' sugar in the pink region here. On the 5' end, the 5' end phosphate is attached. This is kept in a horizontal manner, and this is drawn in a vertical manner. It's just that this has you just move this up straight, so it becomes like this. You are drawing it, and from the 3' end, this O, this attach. This is a 3' end. So, and this bond which is formed over here, it is known as the phosphodiester bond. It joins with a next nucleotide which has G or any, it can be T or any other base in its place. This is a phosphodiester bond. Another phosphodiester bond. So always you can see that the linkage, the link, the phosphodiester link, it is between the 3' end and the 5' end. So always there is a 5' to 3' link, the bond. This is this bond is known as the PDE or phosphodiester bond. There is a question: what is the difference between peptide bond and phosphodiester bond? It can be asked. So this is a phosphodiester bond. And a peptide bond is something you read in biochemistry, amino acid. So this is how one question comes, that is Draw the structure of a DNA including 3 nucleotides. So this is a perfect structure. This is the first nucleotide, second nucleotide, first nucleotide. This is the second nucleotide, and this is the structure. So you simply draw this and get your full marks. So names of the base.
ভার্টিক্যালি আঁকতে হবে না হরাইজন্টালি আঁকলেও হবে?
হরাইজন্টালি তুমি আঁকলেও হবে, হ্যাঁ। হরাইজন্টালি আঁকবেটা কি করে? তোমার তো এর তলায় তুমি লাগাতে চাইছো, ওই জন্য এটা ভার্টিক্যালটা কেন করলো? ভার্টিক্যালটা করার কারণটা হচ্ছে এই ডায়াগ্রামে এই হরাইজন্টাল হলে এর তলারটা আঁকবেটা কি করে? এখানে হরাইজন্টাল হলে তাহলে তো তা তোমাকে সব প্রত্যেকটা জায়গায় এই বন্ডটাকে এরম করে বেঁকিয়ে এরম করে সব বেঁকিয়ে আঁকতে হবে। টু মেক ইট স্ট্রেট ওরা এই এইটা এইভাবে করেছে, বুঝতে পারলে? উপায় নেই, মানে তোমার করতে গেলে বিচ্ছিরি বাজে লাগবে দেখতে। দিস ইজ দ্য ইজিয়েস্ট ওয়ে ইউ ক্যান ড্র, দেয়ারফর দে হ্যাভ মেড ইট হরাইজন্টাল। নয়তো কি হচ্ছে, তুমি এখানে একটা করবে, এইখানে একটা ফসফোডাইএস্টার বন্ড করে আবার আরেকটা করবে, আবার আরেকটা এরকম করে এরকম বেঁকে যাবে। অনেক জায়গায় অনেক বইতে আঁকে ওরকম করে। ইনফ্যাক্ট এই বইয়েতেও বোধহয় পরের স্ট্রাকচারগুলোতে আঁকবে। So that this was a DNA nucleotide and this is an RNA nucleotide. You just simply have a OH instead of H. So I would recommend you please revise this table, maybe make a print of this and keep it in your room. To what are the names? Nucleoside হচ্ছে deoxyribose plus base, you call it as deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. Nucleotide, it has got a phosphate, you call deoxyadenylic acid or deoxyadenosine monophosphate, DAMP, or DGMP or DCMP, DTTMP. Some places to confuse you, draw the structure of deoxyadenylic acid, and now you get confused. What the hell is deoxyadenylic acid? It is nothing but you are drawing the DAMP. MP means monophosphate, single phosphate. You are drawing this. This is nothing but the deoxyadenylic acid. You can call it as deoxyadenosine 5' monophosphate or deoxyadenosine monophosphate, some people call. So just don't get confused by the nomenclature jargons. This is a sort of thing which you people keep in mind about the structure. So RNA, you have nucleoside, a ribose sugar instead of a decribose, and a base. So this is like this. Here you are calling it deoxyadenylic acid, here they will call it adenylic acid. So nucleotide ribose plus base plus phosphate group. It's the same thing, and only in this case in RNA, remember there is no thymidine over here, or nothing like a thymine base is not present in RNA, and no uracil in DNA. Others are common. So this essentially you talk about the structure of DNA, RNA, or a DNA strand, or a RNA strand. RNA strand, DNA strand, they more or less look similar. Now let us go to the double helical structure of DNA, or you can call it as the second part, Watson and Crick model. Watson, Crick. Watson and Crick take the major credit for all these things, but I would like to remind you, here's the picture. Yeah, Watson and Crick in the younger days. Crick has expired, Watson is still alive, something like 80-90 years old. Watson is there, and Francis Crick has expired, and there were other people also. An X-ray, Rosalind Franklin, this lady, the major work. The disaster, the sad thing was she died of cervical cancer at the age of 37 only. And so when she had done all these work of the most of the X-ray diffraction work on the data from the basis data of which I'll just show the picture of Rosalind Franklin. This is Rosalind Franklin. She died at the age of 37. So mostly she found out all the results from a technique known as X-ray diffraction, and this X-ray work of this X-ray diffraction work was discovered by Maurice Wilkins. He also got the Nobel Prize in this part. He deciphered all the unpublished works or all her notes, Rosalind Franklin's notes after her death. They were and he compiled these data, and of course he gave some of his own findings also, fine-tuned it. So Rosalind Franklin actually did all the raw work. All the raw work was detected, and so Maurice Wilkins analyzed it. And another person was, I don't know if a picture of Chargaff there, Erwin Chargaff. I don't know if his picture is there. He also found he had also a new set of data, and that is known as base composition study of Erwin Chargaff. What Chargaff did, essentially, he found out that he compared the purine and pyrimidine base compositions of several organisms. They isolated DNA from several organisms. He hydrolyzed the DNA of a number of organisms and quantified the purines and pyrimidines released. His studies show that 50% of the bases were purines and 50% were pyrimidines. So you know what are the pyrimidines? Adenine and guanine are the pyrimidines, and cytosine and thymidine are the I've just the other way around. The purines are the adenine and guanine, and pyrimidines are cytosine and thymine. And it was found that the amount of adenine was equal to the amount of thymine. If there was 5 moles of adenine, then there was 5 moles of thymidine in different organisms. So this was a great finding. So the amount of A equal to the amount of T, and the A by T ratio, that is, if divide the amount of A with the amount of T, the ratio is 1. So if there was 5 moles of adenine, you also had 5 moles of thymidine present. So their ratio will always be 1. So and he found the same for the G by C ratio, also 1. And he also found, because the amount of purines equal to the amount of pyrimidine, the A by G divided by C by T ratio is also 1. This is fantastic. So let us look at table 2. Let's find out table 2. This is the table 2. He found out in human sperm, corn, Drosophila, Euglena, all the data is given. You see the amount of percentage of base. He found out adenine 31% in human sperm and thymidine 31.5. This is more or less the same. In G, he found it out like this. And the ratio of A to A by T, it was something like, it's never exact. So he found out the amount of adenine equal to amount of thymidine, and amount of guanine is equal to the amount of cytosine. This data and an X-ray diffraction data. This is how an X-ray diffraction is done. You release an X-ray from a source. You keep your DNA sample over here, and you have a photographic plate. So you have, say, the DNA molecule over here. I'm just drawing hypothetically, and the X-ray comes and hits it. So depending upon the structure, the X-ray will get diffracted. It will go another ray may go in this direction, and on the photographic plate you get this data. Now after you interpret this data, and then you get this structure back. It's more or less something like you release a light you stand in front of that light and see your shadow, and from that shadow you try to reconstruct how you look like. But in a shadow there are a lot of places where there is an opaque region. Here in DNA you have lot of spaces in between. The double helix there, in between there is spaces through which the diffracted light goes straight through. So from this, essentially what you can get, you have a 3-dimension structure pasted in a 2-dimensional structure. So you have a photo of basically the DNA which is coming out through X-ray. Like you are, for example, you have some broken bones. You go to the doctor. Doctor suggests an X-ray. When you go to an X-ray, they place your leg on a photographic plate and they release X-ray. The X-ray passes through your skin, passes through your bones, and on a photographic plate. On the photographic plate, it will show. The bone will be shown, and on that bone the doctor can interpret from that data that your bone is broken. Similarly, from this X-ray, the way the X-ray pattern fell on this plate, it was determined the DNA, what can be the structure. So taking into account this particular X-ray diffraction data from Rosalind Franklin and Maurice Wilkins here, and Chargaff's that amount of A is equal to amount of T, and amount of G is equal to amount of C, and their ratio is the same. They found out, the Watson and Crick's DNA model was constructed in 1953. So Watson and Crick's DNA model in 1953. Just let us look at Watson and Crick once more before we go, because these people are considered legendary. However, I consider the work that the DNA structure is very legendary, but giving Watson and Crick the sole credit for DNA discovery, I think that is very wrong. This lady died, and dead person is not given I don't know if a dead person is not given a Nobel Prize. You have to be living in order to get the Nobel Prize. So since Rosalind Franklin was dead, she was not given the credit. So Rosalind Franklin's work was very important. So, and of course one name is added, Maurice Wilkins' name is added, who integrated Rosalind Franklin's data. So our Nobel Prize system has got lots of flaw. One big flaw is our own scientist Jagadish Chandra Bose. His papers were stolen in Switzerland, and it was somebody called Marconi published this data later. It was found out later, of course, nowadays after 100 years, after 120 years after Jagadish Chandra Bose's legendary work, now they are accepting that no, Jagadish Chandra Bose definitely in his Presidency College, Calcutta University laboratory, he did conducted experiments on radio much earlier. So this was one big failure of the international system, a very important scientist who did not get the Nobel Prize. Even 100 years after Jagadish Chandra Bose, there was another scientist, famous scientist, the name was Dr. Subhash Mukhopadhyay, and from his life, several cinema has been made, Ek Doctor Ki Maut. He was a doctor, and it is very sad to know he was the first person in India to make a cheap, a very inexpensive technique, way of in vitro fertilization, IVF. So Dr. Subhash Mukhopadhyay was the first thing, and at that time the ruling communist government at that time, they did not want in any way Bengal to develop. They wanted to only to control Bengal, or their philosophy, ideology was, I don't know what it was. Basically they started the destruction of Bengal, and this 34 years they destroyed everything of Bengal. And so this Jyoti Bose and his goons, they did not allow, they told that Subhash Mukhopadhyay's work is not good, it is fake. He was told that you will be removed from Calcutta Medical College teaching post if you publish your work or give your paper anywhere in the world. He was prevented from appearing everywhere. And not only that, an ad hoc committee, he was a gynecologist consisting of some CPM backed doctors who were eye specialists. They told that this type of work is not possible, it is fake. But Durga was the baby, IVF baby. It was a Marwari lady. They wanted to keep her identity secret because it was a very conservative Marwari house. So Durga gave the name Durga. That this was a successful. That was not a real name actually. Gave her name Durga to this IVF baby which was born. So he was prevented. 14 days later, in UK, another scientist, he found out that he did this independently. He found out the technique of IVF independently, and he got the Nobel Prize. And Subhash Mukhopadhyay, and he was insulted like anywhere, like anything inside in India. Not in India, in West Bengal, specifically in West Bengal, the communist government of West Bengal, I'm talking. He was insulted like anything, and he was made a fake. He was disgraced. There was even a talk of his medical degree being taken away. So he could not take all this humiliation. He committed suicide in 1981. So this is how our India or our West Bengal treated our scientists. So it is a very sad story. From his life, Ek Doctor Ki Maut cinema was of Shyam Benegal was made. Now Subhash Mukhopadhyay got a recognition in 1986. Somebody from the south, he suddenly found that he also came to across another technique of IVF, how to make in vitro fertilization. And this in vitro fertilization gave a lot of hope to lots of childless couples who were unable to have children. So he was he gave lot of hope to a lot of couples. It's not a very expensive, it's expensive nowadays, but it was so expensive, so they devised a cheaper technique. And now this scientist suddenly one person from Calcutta wrote to this scientist after his publication of his work that there is another work in Calcutta of a late doctor known as Subhash Mukhopadhyay. His work was he worked on this. So he came to Calcutta, found out this data, and then he was an honest person. He told that it is not me who did it first in India. It was Subhash Mukhopadhyay who did it first in India, and then it was given to the International Council. But at that time he was dead, and dead people cannot be given Nobel Prize. So the communist government, they not only destroyed everything in Bengal, they even destroyed intellectual talent. And still these communists, they walk around in this place, and they are called liberals. So their government did all these things, and after this they speak about being progressive. They should die of shame. Go to left or go to right. Life is balance. You walk straight. You have to balance on everything. You need a balance of left and right. Anything, you go to any philosophy in a radical side, left or right, you are a fanatic essentially. So to live a normal life, you need to have a balance. সব কিছুই মেপে চলতে হয়. So anyway, let's get back to our studies after this sad story of Subhash Mukhopadhyay. So next, what happens? So Watson and Crick, Rosalind Franklin, it was an error, like I told. So Watson and Crick get the whole credit. Of course, they did work. It's not that they got it for free. They compiled all the data together and made the wonderful structure, the DNA structure which you see here. This famous structure of the DNA molecule, and this structure where they pose. This famous structure where this is basically for the photograph they made this model. So it's not an exact life, it's just a posing. In those days these photographs were a very important thing which we did not have videos at that time. Nowadays we are taking a class via videos. Photograph is nothing. Everywhere you need photographic proofs. So this is the structure which they came up. This famous structure, Watson and Crick structure. So what are the basically postulates of the Watson and Crick model? The DNA molecule consists of two polynucleotide chains. So two polynucleotides. This is one chain of polynucleotide. as I shown in the earlier picture, three nucleotides attached. I saw one DNA strand of DNA. So maybe over here there are 3 nucleotides attached. The 3 nucleotides are over here. So like this, that's a huge polynucleotide chain wound around each other in a right-handed double helix. Right-handed, I don't know what, it's in the clockwise manner if you turn parallelly. That's in a right-handed manner you go. So you have it, and 2 strands like this, they are wound to each other, and the 2 chains are anti-parallel. So one side, if it is 5 prime to 3 prime, one strand, 5 prime is where the 5 prime carbon phosphate is, and 3 prime is where the 3 prime carbon is. One side is on the top 5 prime. After many nucleotides, the ending is another 3 nucleotide, which is the 3 prime end. So one is 3 prime there, one is in this direction, and the other is in the opposite direction. This becomes 5 to 3, the opposite direction. So they are anti-parallel. Right, and the sugar phosphate backbones are on the outside of the double helix. The sugar phosphate is on the outside and the bases are inside. When it's drawn, the bases are in the inside. I will show the picture, you will see. And A is opposite to T and G is opposite to C. From Chargaff's rule, number of A equal to number of T. From that they derived, so that is A is opposite to T and G is, after of course integrating it with the DNA, the X-ray diffraction studies, and taking these together, they made this, they compiled this data and made a model. So with this is the molecular model of this. You see, A is opposite to T, A is opposite to T, G is opposite to C. So here there are at least 5 nucleotides in one turn. There is one region, this big region which is formed here. This is known as the major groove, and opposite to the major groove is another groove, the minor groove. This distance is 1 nanometer. Between each base, between this to this, it is 0.34 nanometers. So in one complete coil, from here to here, this is 3.4 nanometer. This is the Watson and Crick's model, and this whole thing is one side is 10 nanometer, the whole 1 nanometer. So the whole DNA is essentially 2 nanometer thick. It is 2 nanometer thick, and the strands are anti-parallel. So see, this is the 5 prime phosphate, the ribose deoxyribose sugar attached to this base T. This is the 3 prime, this region, this is the 3 prime. Again, this is the 5 prime carbon. So 3 prime to 5 prime, this is the 5 prime phosphate, so 5 prime carbon phosphate, and it ends on the 3 prime region over here. So, and it is opposite. So 5 prime, this side is, in the other way around, it is written 3 prime to 5 prime. So they are anti-parallel. So this is how the DNA structure is organized. So this is the Watson and Crick's DNA. So complementary base pair, Chargaff's rule, what I've explained in this picture. So this is a nice 5-mark question, the structure of postulates of Watson and Crick's model of DNA. So this essentially it finishes this unit, salient features of DNA, RNA, and the Watson and Crick model of studies. So in today's class, I stopped my lecture over here. It becomes an overdose if I go too much. So on I think it's on Saturday, next day I told. On Saturday, we go to the Saturday. We go for the next part. That is the DNA replication. After that, again we will go on to insects and all stuff like that. So gradually I thought that since it started in college, they have started teaching. I've seen Kaushik sir was possibly teaching with from a notebook. I think it becomes a bit difficult for you people to concept, because I will Kaushik doesn't exactly know how to present it properly on through upgrad. So he asked me for help and I could not find time, so I will explain Kaushik how to do it. I will simply tell him that focus your camera on your notebook and you keep on writing, like you know on a blackboard you write on your notebook, and so from that I think you can see it much better, what is like a blackboard writing. So it's not very difficult. He can easily do that. But he asked me how to do it, so I could not find time to make it for him. So I end my class over here. Then we meet on Saturday. Okay. Bye bye then.
Okay, sir.
Any questions by the way? কোনো কোয়েশ্চেন আছে জিজ্ঞেস করো, কোনো কোয়েশ্চেন থাকলে জিজ্ঞেস করতে পারো। আবির বলো কোয়েশ্চেন থাকলে।
না স্যার ঠিক আছে।
ঠিক আছে, আমি ভিডিওটা পাঠিয়ে দেবো
ভিডিওটা
তোমাদের।
দেখবো, হ্যাঁ হ্যাঁ, ভিডিওটা দেখে যদি আবার কোনো কোশ্চেন থাকে তখন
হ্যাঁ
গুগল
নিশ্চয়ই
ক্লাসরুমে জিজ্ঞেস করবো।
হ্যাঁ হ্যাঁ নিশ্চয়ই গুগল ক্লাসরুমে ফোন করে জিজ্ঞেস করে নিতে পারো। হ্যাঁ, ফোন করলে আমি হয়তো মনে থাকবে না, গুগল ক্লাসরুমে দিলে কি হয় আমি তখন আবার ভিডিওটা খুলে দেখে নিতে পারি। ফোন করছি আমি রাস্তায় আছি, তখন আমার হট করে কোন জায়গাটা আমি বুঝতে পারবো না, তো রাস্তায় খুব একটা যাচ্ছি না, অন্য জায়গায় রয়েছি, তো বুঝতে পারবে না। তো ঠিক আছে তাহলে আই এন্ড মাই ক্লাস ওভার হিয়ার। ভিডিওটা দেখো আর এই বইয়ের এইটা পাঠিয়ে দিচ্ছি, এইটা পড়ো। তারপরে আর নোটস ফোটস তো আছেই, তোমাদের পাঠিয়ে দেবো, নোটস ফোটস গুগল ক্লাসরুমে তুলে দেবো আমি। তো ওটা করে নেক্সট ডে আমি ডিএনএ রেপ্লিকেশনটা চালু করব।
ঠিক আছে স্যার।
ওকে ওকে
ওকে স্যার।